So for Grain Rounds today, it's my pleasure to introduce Dr. Mike Kareta. So Dr. Kareta is an assistant scientist in the genetics and genomics group at Stanford Research, and he's been there since 2017, so maybe a little bit more than a year now. He received his bachelor's in molecular and cellular biology from Texas A&M, a PhD in biochemistry and molecular biology from UC Davis, and did his postdoc at Stanford University. So Dr. Kareta's research program focuses on the basic mechanisms which define cellular identity and how identity may be altered to promote disease. His laboratory uses mouse genetics, cell culture, and molecular approaches combined with next-generation genomic and bioinformatics analysis to understand how stem cell genes promote the formation of multiple pediatric and adult tumors, which is going to be the focus of his talk today. He's published in numerous journals such as Nature, Cell Reports, Cell Stem Cell, and JBC, and is currently a project leader on one of our NIH COBRI grants, the Center for Pediatric Research. So it's my pleasure to introduce Mike.
Thank you, Pete. Everyone here okay? All right. So what I'd like to talk to you, well, actually first I should start off, be all proper, nothing to disclose, unfortunately. But what I'd really like to talk about today is this idea of pediatric cancers, and I'll expand into more general adult cancers as well. But when we think about pediatric cancers, these are cancers that often arise very early in development. These are due to developmental processes going awry. Indeed, when you look at the mutation load that occurs in an individual over their lifespan, you can see that very early on the mutational burden is quite low. And as they progress through life, they start to acquire more and more mutations. Now, most of these mutations are silent mutations. They don't really manifest themselves in any sort of phenotype. But some of these are, in the cancer world, what we call driver mutations. And they have the ability to actually induce a tumor. And you can see once those driver mutations are acquired, then the floodgates open up, mutational burden increases, and that's when we start seeing full-blown cancer in individuals. However, what I'd like to remind you is that early on, this burden is light. And this is borne out in the data. If you look at the mutations that an individual might carry and plot these over, these are the results of large sequencing studies where they sequenced individuals over their lifespan. And what you can see is that very early on, this mutational burden is indeed quite light. And so from that, we can infer that since there are few mutations, and these are actually tumor samples, that these are more likely to occur in driver mutations rather than passenger mutations. Therefore, pediatric cancers present a unique window into studying the mechanism of cancer because they're useful in identifying these key tumor pathways, since most likely whatever tumors or whatever mutations they have are in these driver mutations. So one of these key driver mutations that is commonly found in nearly every human tumor is a mutation of the retinoblastoma, or RB tumor suppressor. It was the first tumor suppressor identified. It's inactivated in nearly every human cancer. And it was identified mostly through this one tumor, retinoblastoma, one pediatric cancer. And its canonical role, if you go back to your early Biology 101 textbooks, what you'll see is its role is to restrict or set up this checkpoint regulating the cell cycle. And for decades, that's been the thought, that RB regulates or prevents tumors from forming due to restricting DNA replication. However, what we've seen perhaps in the past 10, now maybe 15 years, is that's not all that RB is doing. It's actually a very multifunctional protein and quite interesting in that regard. Not only does it regulate the cell cycle and help determine when cells will enter senescence or quiescence, but it also plays a role in directing the differentiation of cells. it actually upregulates many lineage-specific transcription factors, and this list has actually grown since I put this slide together, and in doing so can help cells acquire their terminal identity. So when we think about differentiation, we think of cells starting off in this pluripotent, this naive state, where through typical developmental processes, they can acquire a terminally differentiated identity. We used to think that was fixed, that once cells differentiate, then that's the state that they're stuck in. But some incredible work by Shinya Yamanaka back in 2006 showed that by adding in these 4 transcription factors, that the cells are able to reprogram to a state almost indistinguishable from an embryonic stem cell, a state that we call an induced pluripotent stem cell. And I thought going into this project that this would be an ideal system to study perhaps some of these tumor mechanisms. It's not uncommon for pathologists to recognize that tumors carry varying degrees of dedifferentiation. And if RB was playing a role in driving differentiation forwards, might it perhaps play a role in the reverse process in blocking dedifferentiation? Therefore, when it is lost, cells might be more prone to dedifferentiation and tumors might arise. And this was logical because, as I mentioned, RB promotes differentiation. Reprogramming involves an increase in the cell cycle, so there's lots of changes in the cell cycle during reprogramming. And there are some mechanistic similarities between cancer cells and iPS cells, which I won't get into for today's talk. So, right off the bat, wanted to ask, okay, if you lose RB, do cells reprogram more readily to iPS cells? And long story short is yes, indeed, they do. Show that in both mouse and human fibroblasts, when RB is either deleted or knocked down by short hairpin RNAs, you can see an increase in the number of reprogrammed cells indicated by this red dye for alkaline phosphatase activity. When I test for tried and true iPS cells by looking for reactivation of the key pluripotent genes, one of these being OCT4, I could select for cells that reactivate OCT4, endogenous OCT4, and therefore identify these as tried and true iPS cells. You can see there's a very significant upregulation or increase in the number of iPS cells after RB was lost. So RB, when it's lost, you get more iPS cells. You might ask yourself why. And the first question might be, okay, it's just deregulating the cell cycle. The cells are growing faster. If you've got more cells, you get more iPS. And that's what I set out to test. And one of the ways that I did that is, first I had to identify a marker to look at early stage reprogramming, cells that had started to reprogram to an iPS state. And one of those markers commonly used is SSEA1. It's a surface marker that gets turned on about day 4, day 6 of reprogramming. And there are some great antibodies to it, so we can readily identify it by FACS analysis. Then to measure proliferation, at the point of reprogramming, I stain the cells with this dye called CFSE. And it's nonspecific. It's going to bind to the proteins. It's going to bind to the membranes. And then every time that that cell divides, that dye is going to get diluted twofold, which you could then measure, again, using FACS analysis, and look at this dye dilution as a proxy for cellular proliferation. And so what I saw is the following. First off, if you look at this curve right here in gray, these are cells that were serum starved, so they're not going to grow. So you can look at this as your unproliferating baseline. However, if you look in the black or the blue curves, which are the wild type or the RB knockdown respectively, you can see that if you look at the extent of their dye dilution at either day 4 or day 6, that it's almost exactly the same. And so what this indicated to us is that these cells are not growing any faster when RB is lost when they're reprogrammed. So cell cycle is not playing a role in this reprogramming phenotype. So what is? Well, I've mentioned that RB can bind to and regulate many lineage-specific transcription factors. So a good place to look is what genes then might be regulated when RB is lost during this reprogramming process. So I performed RNA-seq, which is just summarized over here in this heat map. But what I'd like to do is clue you into one observation that I made going through this data. And this is using a software algorithm known as gene set enrichment analysis, or GSEA. What GSEA does is it performs a pairwise comparison between any two states. In this case, either the RB wild type or the RB knockdown maps. And it goes through a series of predefined gene sets, which are annotated by experimental data and all put into this one common database. And it looks to see if any of those gene sets favor one condition or another. The genes in this gene set you can see reflected by these little black bars over here. And you can see that they tend to cluster over in this condition, which are the RB knockdown fibroblasts. You might ask, what gene set is this? This is a gene set specific to iPS cells. So right away, this is indicating to us that when RB is lost, this key pluripotency network, this stem cell network, is being perturbed and actually being upregulated by the loss of RB. So that was done in the context of cells that were reprogrammed. 4 factors were present. They were starting to become iPS cells. What about cells where the 4 factors are not present? These are just fibroblasts growing in a dish by themselves without that external pressure to reprogramming and upregulate pluripotency. In that case, what I saw by knocking down RB is, again, a significant upregulation of OCT4 and SOX2, 2 proteins that are key to establishing that pluripotent state. Now, you might ask, is this a direct mechanism? To test that, I use this process called chromatin immunoprecipitation, or as we abbreviate it, CHIP. And what this does is you end up cross-linking your protein of interest to various genes across the genome. You then sonicate this DNA, which serves to break the DNA up into small pieces with their bound protein still attached. Using standard immunoprecipitation techniques, we can use antibodies to pull down our protein of interest and using magnetic beads physically separate them from the remainder of the DNA fragments that do not contain our protein of interest. And then we could purify these DNA fragments and assess the presence of these individual loci either through qPCR or by submitting them for next generation sequencing to look at binding sites across the entire genome. So I did that and noticed that RB was binding to both SOX2 and OCT4. You can look at the intensity of the SOX2 binding by these blue curves. You can see it's binding right at the promoter of OCT4, as well as binding to downstream of SOX2. This might be a bit of a puzzle. Why would it be binding downstream? SOX2 is actually regulated by 2 enhancer regions. One upstream that regulates its expression in neural lineages, and one downstream that regulates its expression in pluripotent lineages, and that's right where I see RB binding. So RB is binding to these pluripotency factors, it's regulating their expression, but how is it regulating their expression? Well, RB is part of this broader protein complex. Many of the members of this complex are involved in setting the epigenetic state of target loci. It can bind to DNA methyltransferases, which deposit a silencing DNA methylation mark onto the DNA. It associates with Jared members or SYN3 and HDAC complexes, which remove activating marks onto the chromatin. HDACs removing these activating acetylation marks, and the Jared1 members, which remove this lysine 4 trimethylation mark, which is also an activating mark. It can also bind to the sub39H member protein, which will put down lysine 9 trimethylation, as well as members of the PRC2 complex, which put down lysine 27 trimethylation, these being repressive marks. So you could see that the combined action of this greater RB complex is to establish silenced chromatin. So to test how RB loss might regulate the epigenetics of these pluripotency factors, what I did, went back to my conditional RB knockout maps, deleted RB by a Cree virus, and then did this chip-seq technique with antibodies to some of these activating marks, acetylation and K4 trimethylation, as well as these silencing marks, K9 and K27 trimethylation. And rather than walking through all the data that we got from this, I'd just like to summarize what I observed. And what I saw was basically a regulation of genes that was split up into two general mechanisms. One subset of genes were the genes mostly associated with the cell cycle, this canonical function of RB. And I observed that most of the genes that were bound by RB and had their epigenetics changed after RB loss, most of that epigenetic change was regulated using at the level of these activating factors. So when RB is present, there might be minimal acetylation, minimal K4 trimethylation at these genes. But then when RB was lost, these genes became strongly activated. There was large regions of acetylation acquired and very large increase of the K4 trimethylation. Now, it makes sense that you would regulate these genes at the level of activating marks because these genes have to be turned on every time the cell is going to divide. And so you don't want to permanently lock away these genes and turn them off. Rather, you regulate them at the regulation of on or not really on. However, the pluripotency genes are totally different. These are genes that, by and large, outside of the early embryo, do not have any use in the adult body. And so these get very strongly silenced by these epigenetic marks, particularly K27 trimethylation. And what I observed was at these pluripotency genes, certainly when RB is present, it's present at these genes. It recruits EZH2 and HDAX, these two members that I showed that repress the chromatin. When RB was lost, we lost recruitment of EZH2 and HDAX, saw a decrease in the amount of lysine 27 trimethylation, particularly at promoter and enhancer regions, and also started to see an increase of these activating marks as well. So to summarize this part of the talk, I'd just like to remind you what we went over. We saw that RB loss can increase the efficiency of IPS reprogramming, that this occurs in a cell cycle independent manner, and that it represses these core stem cell genes. And I didn't have time to show this, but it also represses their downstream network as well. And so it looks like RB is this general protein that plays a role in shutting off pluripotency, in keeping, preventing dedifferentiation in cells. Now, who are these pluripotency factors? As I mentioned, they're transcription factors, but they're a unique type of transcription factor, which I'll further explain. They're known as master regulators. And I'd like to focus in on SOX2 as one of these, which is a well-studied master regulator. And it's a master regulator of embryonic stem cells. As I mentioned, these pluripotency genes normally don't play a role outside of the early embryo. SOX2 is a bit of a bit unique in that regard, where it does play a role in some tissues of the body, where it can also be a master regulator of neural stem cells and neuroendocrine cells as well. So what do I mean when I say master regulator? Back in '57, Conrad Waddington described epigenetics and their role in regulating differentiation as this landscape where a marble would roll down these little valleys, and due to the gravity, if you will, find one path it would go down, and essentially, once it acquires terminal differentiation, be stuck at this lower state. And it makes particular choices as it might migrate down these valleys. You could think of these pausing points as what you might call an adult stem cell, or a tissue-specific stem cell, on its way to becoming a fully differentiated cell. But what makes these decisions at these particular branch points? What makes this marble maybe take this path and not another? Well, those decisions are made by transcription factors. And so these are the genes that play a role in establishing or regulating cellular identity. If you want to take an example of differentiation, you might look at, say, blood cells, erythrocytes. What are the characteristics that give an erythrocyte its identity? It's got the ability to transport oxygen. It's got a very unique shape to help it transfer, to move through small capillaries. Along with that, it's very flexible, so it can find its way through them. Has a complete lack of a nucleus, lack of DNA, which is quite intriguing. And you have to have a lot of them, right? So these are some of the characteristics of how you might define an erythrocyte. Well, if you look at it, just described, here I'm just going to show it as, say, a series of dominoes. Kind of like Waddington's landscape, but I'm going to change it up on you. Where these dominoes can fall, and then at the end, they'll acquire particular identities, these traits that define these particular cells. And you can see at these individual branch points are these decisions that help it acquire these different identities. This is the function of the different transcription factors to regulate these characteristics. Now, I'm sure everyone's eye right away went to this one domino right on top. And you might ask, well, if all of these down here are the transcription factors that might define particular identities of the cell, who is this one factor up top that sets this whole chain in motion? Well, this is the master regulator. This is the one transcription factor which has the ability to define an entire cellular lineage. Indeed, some of the ways that these are identified is if you overexpress a master regulator in a different cell type, that it will change that cell type and allow it to acquire the identity of itself in that foreign context. So they have that ability to set this entire chain in motion. So when you think about cancer, what are some of the characteristics? You think of out-of-control growth and proliferation, loss of cellular identity, this de-differentiation that is commonly observed. Well, master regulators play a role in regulating all of these different functions, proliferation, cellular identity, which is closely tied to de-differentiation. So what my lab is investigating is, do these master regulators play a role in affecting cancer? So let me step back. I mentioned that SOX2 is a master regulator. It is a master regulator of stem cells, neural cells, and neuroendocrine cells. Most of its work studying it has been in the context of pluripotent stem cells, where it's been found that it can actually play two roles. One of them is in transcriptional activation. And so it can bind to and upregulate these other core pluripotency members, OCT4 I had mentioned, as well as NANOC. And then it can also upregulate a lot of these downstream pluripotency genes, as well as multiple epigenetic complexes which are required for establishing and maintaining pluripotency. But also, interestingly, the same protein can play a role in repressing genes as well, which poses an interesting question when you think about it. One protein binds one place, turns genes on, binds another place, turns genes off. And what it's doing in stem cells is it's binding to these lineage-specific transcription factors and turning them off. So while it's keeping cells in this pluripotent state, it is preventing differentiation at the same time to maintain that stemness. Another unique factor to SOX2 and to master regulators is this this characteristic that we refer to as being a pioneer factor. When you look at chromatin, it's wrapped around these histones very tightly. These histones are packed in very different ways. And for the vast majority of transcription factors, if they find their target DNA sequence wrapped up around a histone, they have nothing to do with it. They can't associate with that DNA because that histone is there blocking their association. However, the small class of transcription factors, these pioneer factors, are still able to bind their target DNA even if it's tightly associated with a histone, even if that histone is further packed into more of a heterochromatic formation where these histones are so tight that normally we think that transcription factors can't associate it with it. These pioneer factors can. And so you'll see that many of these pioneer factors are indeed master regulators. Here's a few of them over here. You could see OCT4, SOX2, KLF4. These are pioneer factors. And as I mentioned, these are 3 out of 4 of those critical reprogramming factors to make iPS cells. Another one that's been well studied is ASCO1, and it's used to make induced neuronal cells. And so why are these cells able to reprogram? Because if they find their target sequences in a cell that is differentiated, so say in the case of a pluripotent cell, you take a differentiated cell, these pluripotency genes are off. I mentioned they're not needed anymore. But these pioneer factors, even if they're off, are still able to find a way to bind and turn them on and reestablish that pluripotency. And so because of that, we recognize that cellular reprogramming, as I mentioned at the beginning, is a possibility. Waddington's landscape is not true in that these cells transit from this embryonic state to a fully differentiated state and only are able to progress in that one direction. We can see through induced pluripotency that they can go back up this developmental hill, as you will. And we can see that they can also transit over these hills, find a new valley, which represents a new cellular identity. So these master regulators, you can recognize that they are incredibly powerful at regulating and determining the identity of a cell. So then, if they're misregulated, can this be bad? Certainly. You can quickly see how a misregulation of a master regulator can rapidly change cellular identity. And when it changes state, might it very well put cells into the state that we identify as cancer. So, when we look at cancer, we often ask ourselves, what role does upregulation of oncogenes play? What role does downregulation of tumor suppressors play? But that is not sufficient in really understanding where do tumors come from. And I can show that to you in one very simple experiment. This is one mouse that we have in the lab we work with quite a bit. It is a mouse that is a conditional knockout for RB. So RB is flanked with these lock sites. When CRE is expressed through this one promoter, it's expressed throughout the entire body. When you add tamoxifen, it becomes localized to the nucleus, and then it excises everything between these two lock sites, and in doing so, deletes the RB gene. So we could take a fully developed mouse, adult mouse, inject them with tamoxifen, and delete RB in the entire mouse. If deletion of this one tumor suppressor is sufficient to form cancer, then this entire mouse would turn into one big walking tumor. But that's not what we see. We see tumors arise in just a few cell types. And so the question is, what makes those cell types unique? Some more direct examples where I can show you how this context matters. Say if you look at lung cancer. If you overexpress RAS, this is one of those commonly looked at oncogenes, what happens is it can be mutated and become constitutively active. And when RAS is, when this oncogenic KRAS is present, you'll see that it typically drives non-small cell lung cancer. This is about 85% of all lung cancers. And it's got a 5-year survival rate of about 21%, which in the field of lung cancer isn't too bad, because I'll spend more time talking about this later on. My lab, we study quite a bit small cell lung cancer. And this is the minority of lung cancers, only about 10 to 15%, but its 5-year survival rate is about 7%. And so it's much worse, but you'll notice that these KRAS mutations are typically found in this one subtype of cancer, but not found in this right here. Why? Well, these two different cancers have two different cells of origin. So this indicates to us that different cells of origin matter in terms of whether a tumor will arise. cell type context matters. But even if you just wanted to clump all cells into one cell type, like in the lung, if you wanted to classify them as basal cells, or neuroendocrine cells, or ciliated cells, are all those cells still equal? No. This is the mouse model, well, a mouse model for small cell lung cancer that is initiated in the mouse due to loss of RB, as well as loss of P53, this other commonly studied tumor suppressant. What we can do is induce these small cell carcinomas by intratracheal intubation of these Cre adenoviruses, which in the lung will delete both RB and P53. Now, if you zoom in at the lung epithelium, you'll see many of these different cell types, the Kareta cells, the basal cells, ciliated cells, and these kind of pyramid shapes, which we call the neuroendocrine cells. These are all RB wild type, P53 wild type, but then when you add this Cre virus, they lose RB, they lose P53, and you would say that, okay, all of these neuroendocrine cells, which are the cell of origin for small cell, should become tumors, right? We don't see them. Indeed, if you quantify this, it's been estimated that there are about 100 million neuroendocrine cells in one mouse lung, or 100 million cells in one mouse lung, with 0.04% of them as being neuroendocrine cells. We know that when we add our Cre virus, there are various ways we test this. We like to run controls. That about 30% of the cells actually become infected with this Cre virus. And so we could do the math and we could say that for one mouse, we would expect to find about 12,000 tumors. We don't see that. We actually see only about 100 tumors. And so this is indicating to us that even amongst cells which we might call as having the same identity, there's cellular variability that sets one cell different from another. What might that be? Now, when you look at cell type specific contexts, the cellular variability, this is well studied. You'll see this studied in many of these when you look at what's a cell of origin of a tumor, right? That's really understanding what's the cell type context that is capable of forming that tumor. Or any of these studies that looks to identify cancer stem or tumor propagating cells. This is trying to identify what cell types are capable of forming tumors. But this idea of studying cells at the single cell, what makes an individual cell capable of forming a tumor is really not well investigated. But to go back to the point I made at the beginning, this is well suited for a study of pediatric solid tumors because since these pediatric tumors arise from very few mutations, ones that mostly hit the key pathways for tumor formation, that studying these we can hopefully identify what it is that makes individual cells capable of forming a tumor. And so together, these two kind of identify this classification of the cell that we might call cellular pliancy. This is this recognition that not all cells are equal. No matter how we try to define one cell type and differentiate it from a different cell type, that all the cells in your body are unique. Anyone who's sorted cells by FACS analysis will recognize this. And so there are variances in these populations that can be due to genetic, but most likely due to epigenetic or developmental factors. And it's most likely these that regulate this cellular pliancy. What makes one cell different from another, perhaps one cell more prone to forming a tumor, and one cell resistant to forming a tumor. Bringing this back to this idea of master regulators, who do you think might have the strongest effect in altering cellular identity, cellular plasticity? It's these master regulators. And so the study of RB is unique in this way for us to be able to study this idea of cellular pliancy. Because while I mentioned that RB loss is found in nearly every human neoplasm, RB loss itself only drives just a handful of tumors. So that tells us right there that RB is subject to the varying pliancy of cells to regulate the capacity of forming tumors. When RB is lost, it typically forms tumors in the pituitary, obviously retinoblastoma of the eye, small cell lung cancer, osteosarcomas, and some pancreatic neuroendocrine tumors. Why that is, we don't really know. People have been studying RB for 40, almost 50 years now, and there's still not a clear answer to this. One intriguing thing, though, that I've noticed is the cell of origin for all of these different tumors. All of them are dependent upon SOX2. Remember I mentioned that SOX2 is a master regulator of not just pluripotent stem cells, but also neural or neuroendocrine cells. And the cell of origin for these tumors are neural or neuroendocrine, except for osteosarcoma over here, which arises from osteoblasts, but osteoblasts themselves are also regulated by SOX2. And so this led us to this quite straightforward hypothesis. Perhaps it's this regulation of RB on SOX2 that leaves these cells susceptible to forming tumors when RB is lost, making them unique to other cells which are able to resist forming neoplasms when RB is lost. So the first way to address this question is we went back to our mouse models. And it's been found very early on that when you delete RB in a mouse, you get multiple tumors, but one of the first that you see, well, the first ones that were observed were these adenocarcinomas of the pituitary. So what I did is I made this model for pituitary cancer in the mice using our Cree-Loxer combination system to delete RB, but also bred in a knockout, conditional knockout for SOX2. So we can see what's the consequence of deleting SOX2 along with RB. Down here I'm showing the pituitaries of these mice. These are all RB knockouts. And you can see mice that have both or even one allele of SOX2 at 9 weeks have started this hyperplasia of the pituitary, which you could see measured over here. However, when both alleles of SOX2 were knocked out, these pituitaries were absolutely normal. If you look at the level of cellular proliferation, again, you could see that when SOX2 is present, this hyperproliferation is observed, indicated by this large number of cells which stain for Ki67, which is a proliferative marker. Again, when SOX2 is deleted, almost no Ki67 staining, quantified over here, almost indistinguishable from wild-type pituitary cells. So this does indeed indicate that loss of RB might induce tumors in these cells that SOX2 is required in, that SOX2 is a master regulator for, because then when RB is lost, SOX2 might become upregulated and allow these various cells to acquire more of a progenitor-like state. So one of the main tumors that we're studying in the lab right now, while a lot of our focus is on pediatric cancers, right now we're doing this larger study on small cell lung cancer. And as I mentioned, this is a very nasty tumor. Lung and bronchial cancers are some of the most common that you'll find in the U.S. population. And while small cell is just a small piece of that, it's got this very low 5-year survival rate. And actually, in small cell, people have started to refer to it as more of a 2-year survival rate because patients die that quickly. One of the reasons why is when they show up in the clinic, most likely they're already late stage at that point, stage 3 or stage 4. They usually have metastatic disease. And usually these patients just have a couple of months left. So it's a very devastating cancer. So to study how, since we know RB plays a role in initiating, how might SOX2 play a role in these small cell lung cancers? As I mentioned before, this is initiated not just by loss of RB, but also P53. We've also added in a conditional knockout for P130. This is a gene that is part of the RB family. And it's not required for small cell lung cancer. However, it does speed up the appearance of these tumors in the mice. So instead of seeing tumors in 1 year, we start to see them after 3 months. So this is fantastic. This makes the folks in my lab very happy. we've also bred in this conditional knockout SOX2 allele just as we did before. And so down here you can see an example of some lungs. These are 3-month-old mice that have had RB or p53 knocked out for 3 months and are therefore null for RB, p53, and p130. You can see when SOX2 is still present, the presence of all these tumors throughout the lungs quantified over here. However, in this pilot study, you can see when we deleted SOX2 at the same time as RB and p53, there's almost a complete lack of any tumors in the lungs, as you can see over here. And what I'm showing you here is actually the result of an early pilot study that we did. My lab has been working on a larger study for small cell, not just looking at 3-month, but also 6-month tumors to see if we age these mice any longer, do we start seeing tumors arise. And we've really been getting most of these mice in the past couple of weeks, so I haven't had time to put a figure together. But I'll tell you the results are quite exciting, completely in line with what we saw in this pilot study, that loss of SOX2 almost completely avoids the formation of these tumors upon loss of RB and p53. So if we look at these established tumors, which we can do in cell culture, these are small cell lung cancer cell lines. These are mouse ones. We also have ones from human patients as well. But generally, in these RB loss-initiated tumors, we see that SOX2 is very highly upregulated. Looking in small cell in particular, if we reintroduce RB just by overexpression through a virus, you can see obviously RB comes up, but then SOX2 becomes downregulated, showing us that this RB SOX2 regulation that I saw in fibroblasts and reprogramming still seems to be holding true in these cancer cells. And then when SOX2 is then knocked down by short hairpin RNAs, you can see that these cells lose their ability to grow rapidly. These are proliferation assays with wild type here in gray and the SOX2 knockdown in blue, and you can see quite rapidly these cells are not growing as quickly in both mouse and human small cell lung cancer lines. So how might we study the mechanism of RB regulation of SOX2 and then SOX2 in the development of these tumors? To do that, we really want to look at the very early window of tumor formation. 1 day after loss of RB and p53, 2 days, 1 week, 2 weeks, and so on. Now in the lung, that's very difficult to do. In a lot of these tumors, it's very difficult to do, particularly in the lung. One reason is because these neuroendocrine cells are quite rare in the lung. So is there a way that we can study what role is SOX2 playing in the formation of these tumors from the very early stages? One way we can do that is by organoid cell culture. And we're going to be studying a few of these different RB tumor models. One of these that I showed already is pituitary cancer. Well, these are pituitary organoids that we have established from mice. You can essentially just digest pituitary, and through particular growth conditions, they will grow into these self-organizing pituospheres, they're called. You can see that they express SOX2. And by tweaking the conditions, we can induce them to differentiate. And at that point, SOX2 will become downregulated. So this is a nice system we can use to study these pituitary cancers. To study small cell, we can grow up lung organoids. These are large three-dimensional multicellular organoids that we can grow and derive in culture. And what's important about them is they comprise many different cell types. So they have the basal cells, the ciliated cells. And importantly, they have those very rare neuroendocrine cells in them. In fact, you can tweak the differentiation conditions of these lung organoids to get them to have as many as 30% neuroendocrine cells. Additionally, we can study retinoblastoma as well by the growth of these retinal organoids. Now, these two organoids are both derived from embryonic stem cells. So one of the things that my lab is working on right now is establishing embryonic stem cell lines from our various tumor models. So we can use that as a source material to make these organoids. And then since they came from our mouse tumor models, all we need to do is infect these organoids with CRE. to induce those early processes that happen in the formation of these tumors from the earliest stages. So then finally, the question you might be asking is, okay, SOX2 seems to be required for the formation of these tumors. When you lose SOX2, you almost don't see any tumors anymore. What is SOX2 doing in these tumors to allow them to acquire that state? One of the ways we've been doing that is by using this chip technique in these cancer cells or in primary tumors to identify what genes is SOX2 regulating. Remember, it's a master regulator. What genes is it responsible for regulating, either upregulating or downregulating? And then the next question is, are any of these downstream networks perhaps druggable? Can we use this as a way to bring new drugs into the clinic? So if this is SOX2, up at the top here, can we find perhaps a downstream transcription factor that SOX2 is regulating that we can at least shut off part of these pathways that are required for these various tumors? But ultimately, what would really be the best thing that we can do would be, can we target SOX2 itself, the master regulator? Now, there has been some success in the past few years of being able to target DNA binding factors. And there's actually some tantalizing evidence out there that the SOX family of proteins themselves may be able to be inhibited by small molecules. This is one molecule that was published by this group out at the Genome Institute of Singapore. And while not very bioavailable, is capable of blocking the binding of the SOX family members. And indeed, there are some modifications that they've shown to this that are able to give it some specificity to SOX2 and not perhaps other SOX family members which you might find expressed in the body. So unfortunately, as I said, this is not really a bioavailable compound, so it's not really, most likely not going to be useful in the clinic. But can we use this as a proof of principle to guide some of our studies to create a new inhibitor that may be useful in the clinic? So there's been some structures that have been figured out for SOX2 and some other SOX family members, particularly of the HMG domain. This is that DNA binding domain of SOX2. And what we are doing in collaboration with Meikana's group at the University of Arizona is they're using a process called molecular docking, where in silico, in a computer, they've got a small molecule library, 10,000 small molecules, which they can sit and see computationally, will they bind to this DNA binding domain? And so do all this screening completely in silico to identify some potential candidates that then we can take and put them onto our cancer cell lines and determine, do they indeed inhibit the growth of these tumors? And so this gives us some hope that we might be able to develop some small molecule inhibitors to SOX2 and hopefully shut down this entire network that is going awry that is driving these tumors. So with that, I'd like to give credit to my lab. They're the ones doing most of this work, both Ethan and Ellen and Hannah, who's a talented graduate student, will be joining the lab shortly. I've mentioned that we are in this collaboration to identify these small molecules with the group of Meikana at the University of Arizona, as well as give credit to Julian Sage and Maurice Verning, my two co-mentors at Stanford, who I did some of these early studies in originally. And of course, the Center for Pediatric Research, which is our COBRA grant, which has provided funding for this work. I'd like to thank you for your attention and be happy to take any questions. I'd like to start off, do any of the off-site locations, do you have any questions? No? Well, I'm happy to open up questions. Yes?
Thank you. I have a question about the experiment that you had with the, you would have expected, I don't know, a tumor in every neuroendocrine cell, but instead you just found 100 tumors, which, you know, just 100 in the lungs of the mice. In the cells that were neuroendocrine that didn't form tumors, did you see evidence of apoptosis? Are these cells just dying?
No.
What are they doing?
So that's something that's really intriguing, and I didn't have a chance to explain that further. That's going to be a whole new branch of investigation of my lab. What made that 10% of these neuroendocrine cells unique and capable of forming a tumor? Through some collaborative work that I did back at Stanford, particularly with Mark Krasnow's group, we did single-cell RNA sequencing of wild-type neuroendocrine cells in the lung. And looking in that data, I saw that about 10% of these cells showed SOX2D repression. I'm not going to say it's full SOX2 activation because these are differentiated cells. At that point, SOX2 has already been turned off. And the levels that we saw were not at the levels I would see when SOX2 is on. But by no means was it off. There was these low levels of expression that were present. And so what I think is happening is that this minority of SOX2 repressed cells are the cell of origin for this small cell lung cancers. And so we are right now breeding up a bunch of different mouse lines where we can do lineage tracing, where we can permanently label cells that are SOX2 repressed at the time of tumor initiation and determine if those indeed are the cells that successfully form a tumor. And if we are able to track that down and show that, yes, indeed, these are the cell of origin, the next questions we'll be asking are, what made these SOX2 repressed cells in the first place? Are they a unique type of perhaps developmental intermediate between these neuroendocrine progenitors and the fully differentiated neuroendocrine cells? Are they a set of cells that exist in the lung normally to perhaps help with regeneration and repair, which has been observed in a few other different tissues? Or perhaps is this just a stochastic state of the cells as they grow, their epigenetics are in flux, and sometimes they transit into these states and out of these states. And so we'll be studying those different models for where those cells might arise, and then also step back and look at what are the developmental processes or environmental processes that might favor these cells existing in the SOX2 repressed state. Small cell lung cancer has a very strong 90% association with smoking. Might these external carcinogens play a role in increasing the number of these SOX2 repressed cells? We don't know, but these will be some of the lines of investigation that we'll be following in the future.
What, if anything, is the consequence of modifying the SOX2D in healthy cells, particularly in young children?
So, like, why would we want to?
Well,
What's the consequence of it?
yeah, I mean, SOX2D has a function in healthy cells.
Yes.
And if you're repressing that to keep them from becoming malignant, what would be the consequence of that in healthy, non-malignant cells?
Yes. So that is obviously one of the big concerns as we're performing the search to identify other small molecule inhibitors for it. As I mentioned, SOX2 is still expressed in, say, neural progenitors. So there might be some developmental effects in the brain if people are treated with these SOX2 inhibitors. I'll say the first thing that we're doing is, while there's some structural information, there's for I think five of the SOX family members, there's 18 members of the SOX family. And so there's structure information for 5. We're working on purifying this HMG domain for all the SOX family members. So that way when we do this molecular docking approach, we can try to be quite specific and identify something that will be able to target SOX2 itself and not all of these other SOX family members which are working to maintain other cellular lineages throughout the body. In terms of SOX2 itself, like say in the neural stem cells, not only is SOX2 expressed, but also SOX1. And obviously we will have to test this in extensive preclinical models, but what my hope is that if we are able to inhibit SOX2 and say not SOX1, might SOX1 be sufficient to maintain normal homeostasis in the neural stem cells, whereas these neuroendocrine tumors seem to be quite unique for SOX2 and not the other SOX family members. So perhaps this larger SOX family might actually help us, because we might find redundant function in these other family members and might mitigate some of these effects by adding a SOX2 inhibitor.
Thank you.
Thank you very much, Mike, for that excellent presentation. What you showed us mostly was mouse data. As we all know, mice are not human. So do you have, or has anyone else done work with cancers that arise in humans and evaluated whether these genes are upregulated, downregulated? What's happening in those tumors actually in humans?
Yeah, so SOX2 is upregulated in human retinoblastoma, pancreatic cancer, small cell lung cancer. In fact, there was, back in 4 years ago, I believe it was, there was a large sequencing study where they sequenced multiple, I think it was about 40 human small cell lung cancers. And they actually found that many of these showed amplification of this region on chromosome 3 that contains SOX2. So there does seem to be some good data that indicates that this RB SOX2 axis is certainly important in human cancers as well. And we're testing this in these human cell lines. So for small cell lung cancer, we have many of these human small cell lung cancer lines that we're testing. For retinoblastoma, I didn't touch on that too much. That's actually an interesting beast, if you will, in that in humans, if you lose RB, obviously you get retinoblastoma. That's how the gene was named. In mice, if you delete RB, you don't get mouse retinoblastomas. And so there's some difference there between mouse and humans. But through a clinical trial that my group did with Mike Dyer down at St. Jude's, we were able to get access to a lot of primary human retinoblastoma lines, which we can maintain as xenografts in the vitreous of the eyes of mice. And so that will be one system which we'll use to study perhaps the effect of these factors or the SOX2 pathway in humans, as well as small cell lung cancers in the grafts.
Thank you. Any other questions for Dr. Kareta? Well, thank you very much again, Mike.